So since we are in the biochemistry chapters, one question I would have for you guys, at least on this topic that's coming up next, is how much has Todd and Sarah covered enzymes and how they work? I would actually think you would have more information in those classes than what they would give in a chemistry class, even if it is called biochemistry. Especially since it's only like half a semester of biochemistry and half a semester of organic. Did they cover anything about them?
We did that last time.
So that's a Bio 111 topic is what you're telling me? Okay. I don't know how they divide up material in the biologies. I mean, I know anatomy and physiology, the anatomy part is perfect. It makes perfect sense what you're studying. Physiology is how things function though, isn't it? And so I could see covering enzyme regulation in that class. I could see it covering enzyme regulation in microbiology even. I don't know if Todd talks about it there or if he assumes you've taken Bio 111. Okay, so what we want to look at today is how enzymes function. And then we'll also look at how we can control them. And so remember, enzymes, and I mean this is a non-scientific definition, but I mean if we're trying to get a visual picture, they're large, complex molecules that are often thousands of amino acids long. And that can vary. I mean, they can vary from as little as a hundred to as many as several thousand. The thing we really want to realize is that when we even say complex molecules and we say they're thousands of amino acids, each amino acid is made up of tens of atoms, right? So we're talking 20, 30,000 atoms potentially in a construct like this. And so they're very difficult to understand. We've sort of gotten a little bit of a view of them is that we've looked at how, secondary and tertiary structure lead to kind of specific shapes for the molecules due to those interactions. And so we realize that enzymes have a very specific shape that's unique to each enzyme. And so they lead to specific shapes, and they're, I don't know, maybe unique's kind of pushing it. For some enzymes, if you change a single amino acid in them, you can render them non-functional. Most enzymes are fairly robust, meaning if you change a single amino acid, yes, you're going to affect how well they function, but you may or may not, completely break their functioning. And so what we have to realize is that they are big and they're complex, and so we're not going to be looking at any detailed, actual structures of them, even though nowadays we actually can figure out the real three-dimensional conformation of proteins. Proteins used to be like little black boxes, say 20 or 30 years ago, where they simply were required for life to occur, but no one really had a very good idea of how they function, or even if they could describe how they function, they couldn't tell you what structure led to that. So a lot of these theories are very general in nature, but as we progress in our ability to measure and to understand these enzymes, they're filling in more and more of the details on it. So having a good grasp on the vocabulary is kind of what we're aiming at. So the first thing we want to look at is the active site. So even though we have a very large, complex molecule, the active site is usually a very small area on an enzyme. It usually is approximately 1 to 5% of the total surface area. And this is the actual region where catalysis occurs. So this is the area where the reaction is catalyzed. And it's generally very specific for one specific molecule. And so my way of drawing or thinking about enzymes is I usually think about them as something like this, where that little triangle represents the active site and the square block represents the rest of the enzyme. Now realize that it doesn't look like a square block and it doesn't have a little triangular shape. They have very complex shapes, and even when you kind of, I guess you can still see under that yellow box. I wonder if that shows up in the recordings or not. Since it's not on my computer screen, I'm guessing not. So remind me if it is or not, okay? No, I should say, remind me if I put too much stuff under it. All I did was draw a little arrow here that said active site. And I guess you still can't see it. I'll just, oh, there you go. So, this is where, when we say it's specific for one specific molecule, for instance, in our carbohydrate example, that's an alpha-1,4 glycosidic bond versus a beta-1,4 glycosidic bond. It's so precise, so specific for only one thing. Or it can also tell the difference between D and L isomers. The molecules that lead through glycolysis with, glucose, or the breakdown of glucose, work for D-glucose, do not work for L-glucose. Pretty specific, especially if I showed you a model of D and L-glucose, I doubt you could tell from the ring structure, or even if I just held it up real quick, which one it is, because you have to be kind of paying attention, or, kind of very specific for that. So the active site is one small area, and probably the earliest model that talked about this was the lock and key hypothesis. And what we'll realize is that as we kind of go forward in our discussion, the model that we use to describe the interactions between enzymes and substrates and the catalyzation of their reactions is going to get more and more complicated. And so what this tries to say is that the substrate is like a key, and the enzyme is like a lock. And simply that there is one substrate for every enzyme. Or that basically there's a 1-to-1 ratio of enzymes to substrates. And I'm not saying that in the body there's a 1-to-1 ratio of the number of molecules. I'm simply saying that idea of specificity where, one is very specific to the other. Now, so this was an early model, and it had one flaw in it, kind of, in that it assumes sort of a rigid catalyst. And so in the early models, the enzyme did not change shape. But, again, that lock and key is kind of like, that might be what you teach a first grader. You say, okay, there's one key and it goes into this lock, and that's kind of how an enzyme and a molecule or a reaction that it catalyzes functions. And then, in 4th or 5th grade, you might say, okay, well, you remember when we told you about that lock and key? Well, it's a little bit more complex than that. Imagine that the lock can change shapes, but that it's still specific for one key. And so that kind of led into what people talked about as the induced fit model. And realize that some of this, in the scientific literature, if you followed it, we might be covering years of scientific research or thought about these matters, and we're covering them in minutes. So what we want you to do is have just a general idea of what each of these models, sort of its main point, and then maybe either its flaw or what led us to change something. And so like that lock and key model was great in that it talks about the specificity of the reactions, but it doesn't talk about what actually occurs. So the induced fit model basically says that the active site changes shape when a substrate is present. Shape when a substrate is present, to sort of lock it in place. And so what we're starting to get is the idea that, enzymes are flexible. And that there are actually fairly complex changes that can undergo when a catalyst when a substrate binds to it. So what it does is it generally so it sort of locks onto that enzyme and then can catalyze the reaction. And so what we're moving to is the idea that we have what is called a dynamic catalyst. That the process is not static, and many changes occur during the course of reaction. Any changes. And in this case, we can only give you a couple ideas about what sort of, dynamic processes can occur, because this is going to be specific for every single reaction, meaning a reaction that adds a phosphate to a molecule is going to have obviously different things occurring than when, say, a molecule is broken in half or a bond is strained and broken. But the general idea that the catalyst itself, that enzyme is dynamic, and that what we're really witnessing is an entire series of events instead of a single event where somehow the catalyst binds to the substrate and the next thing you know you have a product. We're trying to kind of get a deeper view of that. So kind of the order that we think about things occurring is that certainly the substrate has to enter the active site. And when we say enters the active site, we don't mean that it's like going inside the molecule or anything. This could be just simply, being kind of pushed up against it and, like thinking about docking a ship at a port or something like that. Then the idea is that the enzyme changes shape. And it kind of does that for two purposes. It binds the substrate, and then it strains, or it either, well, here's where it catalyzes the reaction. And there's sort of a number of different ways it can catalyze the reaction. So, well, I'm kind of going a little fast. Let me just slow down for a second. I probably should have just started a new slide since I knew there's going to be lots of stuff under dynamic catalysts. So the way it changes shape and the way it speeds up the reaction then is in kind of two different ways. So we can kind of put this as like an A under there. The idea of proximity catalyst simply says that it holds the substrate plus any cofactors near each other. I kind of like to think about this as if the reaction were just sitting in place taking in a beaker. In order for the reaction to occur, two molecules have to connect with each other. Well, if there's not very many molecules, the chances of them just accidentally running into each other is pretty small. But if I've got a catalyst or a protein in there, or an enzyme that simply says, okay, when I find one molecule, I'm just going to bind it, and then when I find the other molecule, I'll bind it, and then they'll be close enough to react. And that's more likely to occur than two molecules bumping into each other randomly when there's millions and millions of other molecules kind of floating around in your average cell. So proximity catalyst simply says it holds them near each other. And then the other idea is that we have what's called productive binding. This is when only the desired reaction occurs. Allowed to occur. So basically it holds the reactants in a very specific orientation. So again, if we kind of picture this taking place randomly in the body, meaning, here's my one reactant molecule like this, and that's just randomly floating around, tumbling in space. And in order for a reaction to occur, it has to meet another reactant molecule, for instance, exactly head-on. So these need to be a perfect head-on collision. Well, if we just do this without an enzyme, there's lots of ways that they can hit each other. I kind of think about this too as like, yes.
What is R and what is S? Is that reactant?
R stands for reactants, S stands for substrate. Sorry. Yes and no. Generally, the substrate itself is specific, the one specific molecule that we care about. So like, for instance, if we were doing a reaction with glucose, we would say that glucose is the substrate. When I say the word reactants, I'm saying it sort of in a broader sense, in that, for instance, you might need a phosphate group if you're adding a phosphate group to glucose. And so, yeah, that's me being a little sloppy. Sort of when I say S plus cofactors near each other, I could have simply put reactants. And this is where me being a chemist and, you know, using the biology lingo, I tend to get a little sloppy. So, yeah, S sort of stands for a specific substrate. And that's kind of what we would term our main reactant, maybe. And heck, I guess R stands for reactants. And so, for instance, if we had glucose plus PO4, I'd call them both reactants as a chemist, but a biologist would probably call glucose the substrate, and like things like PO4 cofactors or other things that are needed for that to occur. Jasmine?
So is that why when you increase temperature, is that why you increase temperature, the possible proximity catalyst for productive binding increases as well? It's because the molecules are moving faster?
Kind of and kind of not. One of the things that we're sort of glossing over here in this idea of productive binding is that you certainly do need enough energy for the molecules to react. It's kind of like, you know, if two things bump into each other with very little force, not much happens. But maybe if they bump into each other with enough energy, that's enough to break a bond and to reform a bond. And so that idea of productive binding only letting the desired reaction to occur, if we kind of think about that in terms of activation energy, if we're increasing the temperature, we're increasing the number of times the molecules bump together, right? So there's more chance of a reaction. But more importantly, we're increasing the amount of energy they bump together with, so it's more likely for the reaction to occur. And so sort of the effective temperature on a reaction, I don't know if you could lump that in necessarily with productive binding or not. The idea behind productive binding is really that it holds the reactants in the very specific orientation that they need to react. So it orients everything perfectly. For instance, if we look at that glucose plus phosphate reaction, and I can't remember which bond it makes first, if it's the number 4 carbon or the number 6 carbon that gets the phosphate added. I want to say it's the number 6 one, but I could be wrong. Certainly you can add a phosphate group to just about any carbon in that glucose molecule, but the only one that counts, the only one that actually leads to the rest of the reactions occurring is if that phosphate is bonded to the number 6 carbon. So instead of just having a glucose randomly bumping into a phosphate and adding to any one of 6 carbons, if we kind of think about proximity catalysts and productive binding, it holds them close together and then it holds them in the exact specific orientation for that reaction to occur. And so, you know, if this is how it has to happen sort of randomly, what we could say is that an enzyme is going to go like this. It's going to hold it in place. So here's my enzyme. Here, let's draw our enzyme like this. So it holds it in place, and then it holds this one in place. And then the idea is that, for instance, you could think of this like a hinge. When both reaction molecules are in place, it just goes bam and slaps them together. And so that's the idea behind it being a dynamic catalyst, that there's actually changes in the shape or conformation of that catalyst to cause that reaction to occur. And so what productive binding does is it only makes the desired reaction is allowed to occur, and it prevents undesired reactions. And so maybe you could think about it even like a conductor, you know, someone conducting traffic. If you've got the traffic cop out there, traffic moves more or better, and there's less chances of accidents occurring. And so one of the big things today to study is people will study a very specific enzyme substrate pair and try to figure out what physical changes occur in the enzyme to allow it to function. And we'll talk about some more things that occur, meaning, like we said, these enzymes are complex molecules. This active site in the molecule that binds things is only 1 to 5%. So you got to ask yourself, is the other 95% doing something useful or not? Certainly some of the enzyme is simply to hold things in the right place, so it's not as crucial. But you know, there are other parts of the enzymes that sort of have functionality. So those are kind of the ways that we're thinking about how enzymes speed reactions up. They speed them up by making sort of things, oh, here, look, if I looked in my notes, yep, it is the number 6 carbon. So proximity catalyst would say, like for instance in the glucose reaction, here, let's just draw it. So here's an example. So here's my glucose. Good practice, if nothing else, for drawing these molecules. Oops. And for this reaction to occur, you've got an ATP, you've got hexokinase, which is your actual enzyme. You actually have to have a magnesium 2+ ion all floating around in the right space in order to simply add a phosphate group. Oh, I forgot to add the phosphate group in there. No, the phosphate group technically comes from ATP. You guys are familiar with the structure of ATP? This really stands for adenosine triphosphate. And see, that's what's weird about thinking about this reaction and using it as an example. We haven't even talked about ATP yet. That's in the next chapter. But tri means 3, right? So there's 3 phosphate groups on it. So it makes this molecule plus ADP plus a hydrogen. And so the rest of the OHs don't change. And so the idea is that proximity catalyst holds all 4 parts near each other, as opposed to those 4 molecules randomly hitting each other. And then what productive binding says is that only that carbon is allowed to react. Means only the number 6 carbon reacts. Because you really can put a phosphate on any of those OH groups. There's nothing that says you can't do it on the 1, 2, 3, 4 carbons. You really can't put it on the number 5 carbon because it's part of the ring. And so, to me, that's complex. That requires a lot of things to actually occur. If you just threw, you know, glucose, ATP, and magnesium in a beaker, I'm going to almost say that nothing's going to happen. At least nothing very quickly. The second you add that hexokinase in to catalyze that reaction, now you're making it go a billion times faster, or whatever sort of random number there is associated with that. So that's sort of how enzymes function. And then the next thing we want to look at is how do we control the reaction rate, or how do we control enzymes? Because you don't always want an enzyme working at 100% efficiency. Sometimes you want a different biological process to occur. Sometimes you want to speed something up. Sometimes you want to slow something down for a chemical reaction in your body. And so we can kind of talk about how those things work. The first thing we want to talk about is enzyme inhibitors. And these are basically molecules that bind to enzymes and decrease their activity. Now see, this is where Jay, as a chemist, has an argument with biologists. I'm not sure why they talk about enzymes inhibitors and enzyme activators separately. So my theory is we can also just put in the word activator there too. And that's obviously going to increase the activity. To me, the two concepts are the same, with sort of equal and opposite, I don't know if we can say equal and opposite, but with sort of opposite outcomes. And so whether we're inhibiting an enzyme, meaning slowing its reaction rate, or we're actually activating an enzyme and increasing its activation rate, to me, means the same thing. And so, since this is my class, and I'm a chemist, we're just going to call it the same. And there's sort of two ways that they can do this: irreversible and reversible. So basically, every inhibitor or activator, every molecule that is used to control or regulate these enzymes can bind in basically one of these two ways, either irreversible or that reversible. And generally what irreversible is considered is that they bind tightly, and they generally make a covalent bond. Whereas in a reversible one, they bind loosely, so they're fairly easy to remove. And really, to me, an example of that sort of makes that more obvious or something is like, for instance, the functioning of nerve gas. So there's a bunch of different types of nerve gases and toxins and poisons that you can actually have work. But the way most of them work is that they're basically irreversible binders. And so what some types of nerve gas do is they actually make your muscles permanently contract. In fact, now I haven't witnessed this, but I've heard tell that like a death from this type of nerve gas is one of the most painful things possible, because your whole body basically, every muscle in your body contracts. It can't loosen, and so you're in that constant sense of pain, and that you can actually like break limbs and things like that, that the contractions are so intense. And so that's an example of something that would be considered an irreversible thing. Now, they do have antitoxins and things like that for nerve gases, meaning they do have agents that if you inject them quickly enough, they'll actually go and they'll cause it to be a reversible reaction. And so what I would say is that most enzyme inhibitors, there's very few of them that are actually irreversible. Now, it might be irreversible in the human body under normal circumstances, meaning you have to have an outside drug injected, but it's not. And so whether an inhibitor or something that affects the enzyme is reversible or irreversible, those are kind of gray definitions, right? Meaning we'll say something's irreversible, but obviously if you administer an outside agent, it's reversible then in the body. And then the two other designations that they kind of give for that are they can talk about whether they're competitive or non-competitive. And so this is like being able to say you can label every shirt as a specific color, and you can label every shirt as a specific material, meaning these are just descriptors that are used to talk about different types of enzyme inhibitors or activators. So competitive usually means it binds to the active site. And so AS stands for active site. Whereas for non-competitive ones, they tend to bind to another region of the enzyme. And because a competitive inhibitor binds to the active site, it generally looks similar to the substrate or the reactants. So one way that we can control things in the body, probably the earliest mechanism that we've tried to do as scientists, is simply if we know it binds a specific molecule, then we try to test it with other molecules and see if we can fill in that active site. And so if we can fill in the active site with something, then the active site is no longer available to catalyze the reaction. So what this does is this sort of plugs the active site, preventing a reaction. The way a non-competitive one generally works is it changes the shape of the enzyme so it no longer binds. To, say, a substrate or reactant there. We'll even just start using S and R together, substrate and reactant. And what that does is that if we take a look at the concentration of substrate and the reaction rate, is they kind of function in different ways. So the solid line here is going to be a normal enzyme. Meaning that, remember when we talked about enzymes, they tend to catalyze reactions, and that at some point as the concentration of substrate and enzyme approaches 1:1, they can't actually speed up the reaction anymore, and they sort of plateau at a maximum rate. And so this is the maximum rate. And so a competitive inhibitor tends to have a lower the reaction rate at low concentrations, but it still has the same maximum rate. So the red line will be our competitive. And so at low concentrations, it decreases the rate. At high concentrations of substrate, sorry, I should put that in there, it has no effect. And so a competitive inhibitor is important if there's very little substrate present, but if there's enough substrate present so that it fills the active site all the time and the competitive inhibitor can't bind there to stop the reaction or to prevent it from occurring, it has almost little or no effect. And then if it's a non-competitive one, that'll be our green line, it actually has an effect at all concentrations. So this is our non-competitive, and it decreases the reaction rate at all concentrations. And that's because it actually changes the shape of the enzyme. And so it doesn't matter how much substrate is present. Once it's deactivated and can't catalyze a reaction. So inhibitors and activators tend to decrease or increase reaction rates, and they're how we control how an enzyme functions. Now, I won't say a very specific example, because like I said, we're doing all of these as generic examples. If you want, I should remember I can post a link or two to just recent scientist articles where they literally are, discovering where the active site is on enzymes, and they're figuring out what sort of inhibitors or where the inhibitors bind to those molecules. So one way that we can regulate enzyme activity is through what's called allosteric regulation. And so this is a control mechanism. And I shouldn't say, I don't even know if I like the word mechanism. It's a control idea. It's how we believe these things function. So it's a control mechanism for enzymes. And it generally applies to complex or multi-component enzymes. One thing that we should also realize is that enzymes come in a variety of complexities, meaning we can have single enzymes that have only one unit or one piece to them, in which case, allosteric regulation may not occur, meaning you may have to use another mechanism to control their rate by simply inhibiting them with a secondary molecule. A lot of enzymes, remember, if we take a look at their quaternary structure, that's where they can have multiple subunits and multiple parts to them. That's where something like allosteric regulation probably plays a bigger role, in that there's more parts of the molecule that it can bind to. There are two parts, and an enzyme can have 1, 0, or none of those parts, or two of those parts. Enzymes can have 0, 1, or 2 of the features. That's kind of being generic, I guess. What we're really saying is that if it has none of these features, then there is no allosteric regulation, really. It can have one where maybe it's just the inhibitor or the activator, or it can actually have two sites, an inhibitor and an activator site. And the other important part about this is that these are not the active site. And so what we're really talking about is a mechanism for non-competitive binding. And now we're going to get into some uber awesome pictures. So I'm going to go to another slide so I can kind of fit this all in. And realize that I'm kind of simplifying this a little bit in that I'm showing both features at once. So the main thing I'm trying to show here is that there are two different binding sites. And then there's a second part of the molecule, like this, for instance. Oops. Should at least make them different. And how you draw this is, there's a million different ways. And so what we're going to say is that this is an inhibition site, inhibitor binding site. And then, of course, we can also have an activator binding site. And then we'll call this big feature down here the active site. And remember, I even said the active site was a very tiny, small part of the molecule, so I'm obviously exaggerating the size of it. And then the two units, we can kind of talk about this top unit as being what's called the regulator subunit. It's what allows something to be increasing the rate or decreasing the rate. And then this is sometimes referred to as the catalytic subunit. And so what we're doing now is we're taking a protein and saying there's a lot of different features that we can start pointing out. This is beyond, pointing out things like primary structure, which is just the order of the amino acids, or secondary and tertiary structure, which is how the parts interact, or even quaternary structure where there's multiple parts. This is being nice and vague and simply saying that some part of the molecule has a binding site that if a molecule interacts with it, it activates that protein and causes it to react. And that there's some site or some feature on that molecule, probably made up of many amino acids, that can act, if a molecule binds there, it inhibits the reaction. And so then two things can happen. We can have an inhibitor molecule react with it. And boy, did I draw that way too big. So we're going to have to shrink our molecule down so I can fit it in. So the idea is that that triangle, that inhibitor, interacts with it. So we've filled up the inhibitor site. And what that does is that causes a change in conformation or shape of the active site to prevent catalysis. Or basically, it's like we turned off it. Maybe we can think about it as a car. This is a really horrible analogy that I'm coming up with on the top of my head. You can think about it as a car. If you put a low jack or a wheel jack on it, can the car move? Like one of those little parking things that they give you. What do they call those? When they like put the tire clamps on? That would be an example of like an inhibitor, right? That pretty much prevents the car from moving terribly well, right? And of course, we can have the exact same thing. I shouldn't say exact. To me, it's the same thing, even though it's the opposite. If I have, for instance, an activator reacting. So here's my activator. And again, making the picture smaller because we ran out of room. So we've plugged in the activator site. And what that does is it causes a change in conformation to speed up the reaction. And these are all very generic, meaning, you know, some molecules might only have an activator site, some might only have an inhibitor site. Sometimes the inhibitor and the activator site are the exact same thing, and it just is considered an on-off switch. You know, sometimes the active site might be fully active without anything attached to it, and then the inhibitor breaks it. Sometimes the active site might not be, you know, catalyze the reaction unless an activator actually changes its shape. And so, you know, we've got 3 different features interacting with each other. You can probably think up of like 6 different control schemes for it, depending upon how those pieces interact with each other. But we want to keep this fairly generic. And so really the idea is to realize that there are different regions on a protein that control its functionality in a chemical reaction. And that's how the body kind of does this. One other mechanism that's interesting is called cooperation. It's slightly different from what we would consider allosteric regulation, mostly because it actually does kind of bind to the active site. And this generally occurs when you have multiple identical subunits. And the general idea is that if you bind one active site, it causes the other active sites to bind easier. And because it kind of binds to the active site, it's not really technically what they would call allosteric regulation, even if it has the same general principles of it, which is simply binding something to an enzyme causes it to increase. And the classical example of this one is hemoglobin. The activity increases if one site binds oxygen. What generally occurs is that, you know, a hemoglobin binds one oxygen and then it very quickly binds 3 other oxygens. And so that hemoglobins tend to either be full of oxygen or have no oxygen on them. And so they function like that. Okay, so that's sort of one way that we can control the functionality of it, by binding these activator or these inhibitor molecules to the specific regions on the protein. Another way that you can do the same thing kind of is through covalent modification. Covalent modification. And there's 2 examples for this. What we're really doing is that we bind, well, let's just do one. So we bind a functional group. Actually, I don't like that word bind. We add or remove. So this is kind of like allosteric regulation in that, you know, we're going to change the shape or the functionality of the enzyme. It's different in the method in which it achieves it. Allosteric regulation does it by binding a small molecule to it that can kind of come and go, meaning it's probably held in there by like hydrogen bonds or something like this. This tends to actually bind or actually cause a chemical reaction to occur and attach a functional group to it. Probably the most common example is phosphorylation. Phosphorylation. And this is the addition of a phosphate group. So remember, a phosphate group kind of looks like this. to a protein. And it generally binds to serine, threonine, or tyrosine, meaning that for whatever reason, these three specific amino acids is where this happens most often. And what this does is it leads to a change in the charges. Because remember, a phosphate group generally looks like this, meaning it has a bunch of negative charges on those oxygens. And if you think about it, if we write it like chemists do, we would just write PO4 3-, right? Meaning a phosphate ion normally has a -3 charge. And so this, of course, leads to changes in conformation. And this can be used to activate or deactivate a protein or an enzyme. I guess that's probably a better way to say it. So just like binding an inhibitor or an activator through secondary forces like hydrogen bonding or hydrophobic hydrophilic type interactions, this has got the same effect, meaning it increases or decreases the rate at which it occurs. It's just the specific mechanism by which it does it. The second example of covalent modification is called cleavage. Cleavage. And this is when we break an inactive molecule apart into an active component. And an example of this is some digestive enzymes. They can be, for instance, released in the upper small intestine. Now, here's where you guys can probably help me. Intestine. What's the upper one called? Did you guys like divide the intestine into parts? Yeah, I don't even. That's bullshit to me. I mean, why don't they say upper, lower, and middle? That makes sense to me, right? So it's released in the upper small intestine, but is activated by being cleaved. And you guys know what the word cleaved means? Broken apart into two parts, right? Or more, I guess, technically, but two in this case. By being cleaved by another molecule or another enzyme in the lower intestine, lower small intestine. It's one of the things that has always bothered me about some things. So like, let's see if I can remember this. So there's maxillary and mandibular, right? And one talks about the top half of your mouth and one is the bottom half of your mouth or something. I'm sorry, but when did top half and bottom half not work? Maybe it sounds better to say mandibular or maxillary because it's better. It sounds fancier, but I kind of think sometimes we just make up words so that other people can't understand it. It's kind of like its own secret language. Just like instead of referring to teeth by numbers 1 through, how many teeth are there? 26, 18, 19, what? 26? 36? I don't know. How many ever, maybe it is 36 because 1 through 18 rings a bell. So there's 18 on top and 18 on bottom, something like that. But they have to name every damn tooth instead of just saying tooth number 23. That, for me, is a perfectly good descriptor, right? Instead of saying, oh, that's an incisor. Although I guess incisor at least gives you some insight into its function, right? And then if you know its function, you might know its shape. So cleavage is another way that we can affect enzyme regulation by simply changing the from an active state to a non-active state. Let's see, there's 2 topics left, and we'll just make it a long one. That's okay. We'll just add up the time again. I guarantee you that I have to have May, the day right before finals week off, and there might be not this Friday, but the following Friday I might miss that too. So for meetings, so we'll have plenty of time to do that. So the way there's two overall mechanisms that can be discussed. There's feedback inhibition, and you can talk about feed forward activation. So this is taking a step back and not thinking about the mechanism by which it occurs, but the result by which it occurs. And so what feed forward or feedback inhibition generally says is that if there's an excess product, it acts as an inhibitor to an earlier step. In a reaction. So if we think about it, most biological pathways are a series of steps. For instance, A goes to B, goes to C, goes to D, to an earlier step. The key is to recognize that feedback inhibition simply says that something at the end slows something at the beginning. So for instance, this molecule, oops, I have to be careful. So let's use a different color. This molecule acts as an inhibitor for enzyme 1. So if we say that there's different enzymes controlling this reaction, enzyme 1, enzyme 2, enzyme 3, then what happens is that the molecule D actually acts as an inhibitor for an earlier reaction step. And what that does is it simply says, if I've got tons of the product around that I'm not using, I don't need to make any more. And so what it does is it says, let's inhibit an earlier step in the reaction to slow down the process. It would be like if you have a production line that is making cardboard boxes to put eggs in, and you've also got chickens laying eggs, right? Well, if you've got 5 million boxes and like 10 chickens, do you need to make any more boxes? So what happens is, if you've got more boxes than you've got eggs, it tells the conveyor belt to slow down. Now, maybe that's a silly analogy. It's what happens when Jay comes up with funky analogies off the top of his head. Could be, I don't know if this really makes any sense, but feedback inhibition might be after you've had about 6 drinks, do you really feel like having another drink? No, you kind of get dizzy and you have to go to the bathroom. If you're having beer, you have to go to the bathroom a lot. That slows down the amount of beer you can drink. No? I find that when I have to start going to the bathroom every 5 minutes, I just don't drink as much beer. But that's me, maybe. So that would be an example of feedback inhibition. Once you've started to have too much, you tend to slow down. At least normal people do. Okay. Feed forward activation is the exact same way. It basically says that if I have an excess of reactants or products in an early part of the reaction, it acts as an activator for a later step. So if we kind of take a look at that same idea, I've got A, it's turned into B, turned into C, it's turned into D. I've got various enzymes controlling it. What feed-forward inhibition says is that, for instance, molecule A here acts as an activator for this last step here. So molecule A is an activator for enzyme 3. So what it's saying is that if I've got a ton of my substrate present and I really need to get rid of it, and I really need to react it away and turn it into something more useful, it can actually speed up later steps in a reaction and act as an activator. And then once you've used it all up, it no longer activates the reaction, and so the reaction rate slows down. And so we can basically speed up or slow down reactions by having a product early in the reaction or late in the reaction affect an enzyme before it or after it in the chemical reaction pathways. And so that's called feedback inhibition when you want to slow down a set of reactions, and feed forward activation when you want to speed it up. Okay, one last topic and then we're done. And this one is one of those miscellaneous topics that I don't know if it makes sense to do it earlier or later, but we want to look at the effect of temperature and pH on enzymes. And so this is not what we would call a regulatory sort of action, meaning we're not causing an enzyme to function better or worse by controlling it through a specific inhibitor or activator. This is just a general physical process, meaning if I increase the temperature or decrease the temperature, that affects the rate at which enzymes function. And so my favorite picture to draw that kind of captures it is we just look at temperature and we look at reaction rate. What we typically find, and the shape of this curve can vary, but we find that there's an optimal temperature for most enzymes. And that optimal temperature varies depending upon what type of enzyme it is. For most enzymes in the human body, it's 37 degrees Celsius. But for instance, if you go and you look at, say, thermophiles that live in high temperature ocean vents, then their enzymes tend to function most efficiently at higher temperatures. And this occurs mostly because of the change in shape due to how temperature affects secondary and tertiary interactions. So if there's more energy around, it means that the intermolecular forces, I guess we could say this, we kind of used to do this. If we say that the energy is stronger than the intermolecular forces, then they no longer hold a specific shape. It's kind of like, we're kind of saying that, well, I guess we could, this is essentially what we're saying is that they become denatured. But what we're saying is that they're not completely denatured, but they are changed in shape enough so that they're no longer quite as functional. Think about that enzyme site that if it's very rigid, if it has a very specific shape that it binds to, if you change a hydrogen bond in there and that shape changes just a little bit, then it's no longer able to bind the substrate as effectively. Maybe instead of locking it in place, it only goes in there sometimes and can pop back out kind of easily. So this would be at high temperatures, obviously, if we have lots of energy around. And then at low temperatures, it's more of a kinetic effect, is that we simply lack the activation energy for a reaction to occur. If the temperature is too cold, then your body, the enzyme, doesn't have enough flexibility, or it can't generate enough energy to catalyze that chemical reaction. And then we can draw an identical graph for pH, except instead of having temperature on the x-axis, what do you think we're going to put? Yeah. So if we take a look at pH and the activity rate, we get a similar reaction. And again, there's an optimal temperature, or I'm sorry, in this case, optimal pH. And for the body, that's on the order of 7.4 usually. And pH also basically affects both secondary and tertiary interactions, and that it changes how side chains like carboxylic acids and amines form ionic bonds. Oh, I'm sorry, we called those salt bridges if we're chemists. And it can also affect things like the formation of hydrogen bonds, and it can even affect things like hydrophobic or hydrophilic interactions. And really what we can kind of think about it is at the extremes, so basically at high and low pH, we tend to denature. Or we change the shape of the protein enough so that it functions. An example of this is one of the common things to add to soaps nowadays is various enzymes. But depending upon what the soap is used for, you might want to have your enzyme function differently. For instance, if you're doing dishes versus doing laundry versus doing industrial level cleaning, that occurs at different temperatures and different pHs, just like you can have hot and cold for laundry. Now, out of curiosity, does anyone ever do anything with the temperature of their, like, I just leave my washer on 1. As far as I can tell, they used to say that colors would fade in high temperature, so you're supposed to wash them in low temperature, but I think colors are usually good enough now that they don't tend to fade too much. I don't know, do you guys do your laundry at different temperatures? Oh. They used to say don't mix colors and whites. I throw my white socks and t-shirts right in with the colors and haven't noticed anything funky happening. Okay, so let's take a look at the rest of the week to finish up real quick. So we said we're going to have the test on chapter, 28 through 30. So that's lipids, proteins, and enzymes. And I, for sure, if I remember right, put out study guides or study sheets for those, so there's that. I don't remember if I've done enzymes yet. I might have to do that this afternoon. I can't remember if I put it up there or not now. Wednesday, we're going to start in on chapter 31, which is DNA, etc. Tuesday, we have lab and homework 30A and B due. And Friday, I don't know, we'll just lecture like normal.
What if we have a class at noon on Thursday? When we have our test and
So what you're doing is saying schedule the test. Well, what would we do during lecture during lab? That would work if everyone managed to finish the test at exactly the same time.
But the lab time is longer than our lecture.
Right. But what I'm saying is if you could convince everyone to start the test at noon and finish by, what would we have to finish by? 1:30 on Thursday. She's wanting to try to do it. Then maybe you could